is some resources and materials for IB students to help them through topics 1 and 3 of biology, and these are part of the core topics, so this is not additional HL material. And we'll be first discussing in this video the stages of meiosis. Meiosis is a type of cell division that results in the production of 4 genetically unique daughter cells, each containing half the number of chromosomes as the parent cell, so they are haploid cells. Meiosis is essential for sexual reproduction, and it occurs in all sexually reproducing organisms.
So before we delve into the different stages of meiosis, we should have a very brief overview of interphase. So this is the stage in the cell cycle in which chromosomes are replicated in preparation for meiosis. So DNA is replicated during S phase and produces 2 identical strands of DNA. The identical strands are called sister chromatids and are held together by a centromere. And the diagram on the right demonstrates how the 2 strands of DNA become sister chromatids. And it's also important to mention that when drawing sister chromatids, that they should not be drawn overlapping each other. And then here is a very brief overview of meiosis. So yeah, here's a little diagram. So meiosis consists of 2 rounds of cell division called meiosis 1 and meiosis 2, and that can be seen in this diagram as well. So in meiosis 1, homologous chromosomes pair up and exchange genetic information, while in meiosis 2, sister chromatids separate. And then the end result seen in this diagram is the production of 4 haploid daughter cells.
On to prophase 1. The first stage of meiosis is prophase 1. The first stage of meiosis 1 is prophase 1. And during this stage, the nuclear membrane breaks down, which envelops the chromosomes, and the homologous chromosomes are now free to move around, and they pair up to form tetrads. This is also the stage where crossing over occurs, where sections of homologous chromosomes exchange genetic material, and this exchange increases genetic diversity in the daughter cells.
And next is metaphase 1. So in metaphase 1 is when the tetrads, which are the groups of 4, line up at the equator of the cell. And then the orientation of the tetrads are completely random, and this contributes in increasing the genetic diversity of the daughter cells. And we will be discussing random orientation more later in this video.
So in anaphase 1, the homologous chromosomes separate and are pulled to the opposite ends of the cell, and each new cell receives only 1 chromosome from each pair.
And then we have telophase 1 and cytokinesis. Telophase 1 marks the end of meiosis 1, and during this stage, the nuclear membrane reforms around the chromosomes, and then the cell undergoes cytokinesis, which is the splitting of a cell into 2 daughter cells. So now each daughter cell contains only 1 chromosome from each homologous pair.
And then in the beginning of meiosis 2, which is prophase 2, the process is similar to mitosis, and in prophase 2, the nuclear membrane breaks down again and the spindle fibers form.
And similarly to metaphase 1, this is the stage in which the chromosomes once again line up at the equator of the cell, as seen in this diagram, except this time it's happening in 2 different cells simultaneously.
Then in anaphase 2, the sister chromatids separate and are pulled to the opposite ends of the cell.
And lastly, we have telophase 2 and cytokinesis. Telophase 2 marks the end of meiosis 2 and the entire process of meiosis. So the nuclear membrane reforms around each set of chromosomes, and then the cell undergoes cytokinesis, splitting into 4 daughter cells. Each daughter cell contains only 1 copy of each chromosome. It's also important to mention that both telophase and cytokinesis happen simultaneously. So as the nuclear membrane reforms, a contractile ring will be forming in the center of the cell in order for it to be separated.
And so to summarize, meiosis is a type of cell division that produces 4 haploid daughter cells, each containing half the number of chromosomes as the parent cell. It consists of 2 rounds of division, meiosis 1 and meiosis 2, and meiosis 1 involves the pairing of homologous chromosomes and crossing over, while meiosis 2 involves the separation of sister chromatids, and overall, meiosis is a crucial process for sexual reproduction.
And now we have Mendel's law of independent assortment. So this is one of the fundamental principles of genetics that explains how genes are passed down from parents to offsprings. This is also a very important part of the syllabus. So this law states that the inheritance of one gene does not affect the inheritance of another gene located on a different chromosome, meaning that the distribution of alleles for one trait is independent of the distribution of alleles for another trait. And to further understand this law, it's important to understand a few key terms. So a gene is a segment of DNA that contains the instructions for making a particular protein or a trait. An allele is a variant form of a gene that can produce a different version of a trait. Chromosomes are structures that contain many genes, and humans have 23 pairs of chromosomes. So during sexual reproduction, each parent passes on one allele for each gene to their offspring. So, for example, a parent might pass on the allele for blonde hair or dark hair. So when these two alleles from the parents come together in the offspring, it determines the trait that the offspring will have. So there's an experiment which is called Mendel's pea plant experiment, which shows that the inheritance of one trait, such as flower color, did not influence the inheritance of another trait, such as plant height. He discovered that genes for different traits are inherited independently of each other as long as they are located on different chromosomes. So prior to Mendel's discovery through his pea plant experiment, many biologists of the time believed that the traits which an offspring would inherit is a result of blending the traits of each parent. However, when Mendel cross-pollinated one variety of purebred plant with another, these crosses would yield offsprings which looked like one parent plant, not a blend of the two. So for example, when he crossbred between plants which would have purple flowers and a plant that had white flowers, the offsprings yielded would not have light purple flowers. They would either have purple flowers or white flowers, and this suggests that traits inherited are not a combination of the traits from the parent, and then that the trait which the majority of the offsprings inherit is the dominant trait. So in this example, the majority of the offsprings will have purple flowers, which makes it the dominant trait, and makes having white flowers the recessive trait. And yeah, so Mendel's law of independent assortment helps explain the variety of traits we see in offspring. As it allows for new combinations of alleles to be created with each generation, this law is important in understanding the genetics of inheritance and has been confirmed through further research since Mendel's time. And here is a little chart explaining which trait is either recessive or dominant in pea plants.
The sexual life cycle in genetics refers to the process of reproduction that involves the fusion of gametes, which are specialized cells that carry genetic information from each parent. The sexual life cycle is a complex process that involves meiosis, which we talked about, fertilization, and the formation of new individuals with unique genetic characteristics that aren't just copies of the parent organism. In sexually reproducing organisms, the life cycle begins with the production of gametes, which are formed through the process of meiosis. And meiosis is a type of cell division that results in the formation of 4 haploid cells from a single diploid cell, as we just talked about. And the haploid cells, of course, have half the number of chromosomes as the parent cell, and these haploid cells are called gametes. And during fertilization, a sperm cell and an egg cell fuse to form a zygote, which is a single cell that contains all of the genetic information from both parents. The zygote then undergoes mitosis, a type of cell division that results in the formation of 2 identical cells. As the cells continue to divide and differentiate, they form the various tissues and organs of the developing organism, and each new individual that is formed through sexual reproduction has a unique combination of genetic information due to random assortment of chromosomes during meiosis, and random fusion of gametes during fertilization, and additionally the crossing over that happens in meiosis 1. And this sexual life cycle has important implications for genetics, as it allows for the creation of new genetic combinations and helps to promote genetic diversity within populations, which enables them to survive and adapt to their environment. And this diversity can be beneficial, of course, for the survival and adaptation of the species, as I just said, as it provides a wider range of traits and variations upon which natural selection can act.
So here we have crossing over. Crossing over is a process that occurs during meiosis, where sections of homologous chromosomes break and exchange segments of DNA. So the point that homologous chromosomes exchange segments of DNA is called chiasmata, and this leads to the creation of new combinations of alleles on each chromosome, resulting in genetic diversity in offspring. The process occurs during prophase 1 of meiosis and is influenced by factors such as the distance between genes on the chromosomes and the presence of chiasmata. During crossing over, it can also result in genetic recombination, which can lead to the production of offspring with new and different traits compared to their parents, contributing to the evolution of species by offering variety.
Random assortment is a fundamental genetic process that occurs during meiosis. During meiosis, metaphase 1, homologous chromosomes pair up and separate, leading to the creation of haploid cells, which are, of course, gametes, with half the number of chromosomes as the original cell. The process of random assortment occurs during the first meiotic divisions, where homologous chromosomes randomly segregate into the daughter cells. This means that each daughter cell receives a random assortment of chromosomes from the parent cell, resulting in genetic diversity in the resulting offspring. And the random assortment of chromosomes during meiosis is a key mechanism for creating genetic diversity in sexually reproducing organisms. And this diversity is important for the survival and adaptation of species, as it provides a wider range of traits and variations upon which natural selection can act. And random assortment is influenced by a variety of factors, including the orientation of chromosomes on the spindle fibers and the independent nature of the separation of each chromosome pair. And overall, random assortment is a crucial process in the genetic makeup of offspring, as it generates a diverse genetic combinations that are the foundation of evolution. So basically, even though two organisms have offspring, let's say 10 offspring, each of these offspring will have unique genetic combinations, even though they are formed from the fusion of gametes from these two organisms that have their genetic makeup stays the same.
And now we have random fertilization. So random fertilization is a process that occurs during sexual reproduction in which any sperm can fertilize any egg. The resulting zygote has a unique combination of genetic information due to the random selection of both sperm and egg. This process contributes to genetic diversity as it leads to the creation of offspring with new combinations of genes that are not present in either parent or even sibling. So during fertilization, multiple sperm compete to fertilize a single egg, but only one sperm can be the winner. The chance of any particular sperm fertilizing the egg is completely random and independent of any other egg or sperm. This means that the offspring has a unique set of genetic information. So random fertilization is an important component to genetic variation in populations, which in turn affects the ability of populations to adapt to changing environments. In addition, it is important in maintaining genetic diversity within species, as it ensures that new combinations of genes are constantly being produced. Without random fertilization, populations would be more prone to inbreeding and a loss of diversity, which can lead to decreased fitness and make them more susceptible to disease and environmental stressor.